Hello and welcome into the world of bacteria. Bacteria are single-celled microorganisms that play an important role in our lives and in the world around us. In this video, we will dive into the history of bacteria, their structure, types, classification, advantages, and disadvantages. The discovery of bacteria can be traced back to the 17th century when the Dutch merchant and amateur scientist Anton van Leeuwenhoek first observed single-celled organisms using a primitive microscope. He called these organisms animalcules. Over the years, scientists continued to study bacteria and their role in the world around us. In the late 19th and early 20th centuries, advances in microbiology allowed for the classification and study of bacteria in more detail. The structure of bacteria plays an important role in their survival and interaction with their environment. Understanding the structure of bacteria can help us understand how they are able to perform their functions and how they can cause harm. Cell wall. The cell wall of bacteria provides a protective barrier for the cell and gives it shape. It is made up of a complex sugar-protein molecule called peptidoglycan, which helps maintain the integrity of the cell and protects it from damage. The cell wall is also important in determining the shape of bacteria and can affect their ability to move and interact with their environment. Cytoplasmic membrane. The cytoplasmic membrane regulates the exchange of materials in and out of the cell and controls the movement of substances into the cell. It is a thin layer of lipid molecules that forms a barrier between the cytoplasm and the outside environment. The membrane is semi-permeable, allowing certain substances to enter and exit the cell while preventing others from doing so. Cytoplasm. The cytoplasm contains the genetic material of the bacteria, usually in the form of a single chromosome, and the metabolic machinery needed for the bacteria to perform its functions. It is a gel-like substance that contains enzymes, metabolic pathways, and other cellular structures necessary for survival. Outer capsule. Some bacteria also have an outer capsule, which is a layer of polysaccharides that surrounds the cell wall. The capsule helps protect bacteria from the environment and can also help bacteria evade the immune system by masking their surface antigens. The capsule can also play a role in helping bacteria attach to surfaces and form colonies. There are many different types of bacteria, and they can be classified in a variety of ways based on their shape, metabolic processes, and other characteristics. Some of the most important types of bacteria include Cocci. These are spherical bacteria that can exist singly, in pairs, or in clusters. Examples of Cocci include Streptococcus, which can cause strep throat and other infections, and Staphylococcus, which can cause skin infections. Bacilli. These are rod-shaped bacteria that can exist singly or in chains. Examples of bacilli include Escherichia coli, which is found in the human gut and can cause food poisoning, and Bacillus anthracis, which causes anthrax. Spirilla. These are spiral-shaped bacteria that are often motile. Examples of spirilla include Treponema pallidum, which causes syphilis, and Vibrio, which can cause cholera and other diseases. Mycoplasmas. These are bacteria without a cell wall, which makes them unique among bacteria. Mycoplasmas can cause a variety of diseases, including pneumonia and urethritis. Cyanobacteria. These are photosynthetic bacteria that produce their own food using light energy. They play an important role in the global carbon cycle, as they are responsible for producing much of the oxygen in the atmosphere. In addition to these types of bacteria, there are many other types that can be classified based on their metabolic processes, such as facultative anaerobes, bacteria that can grow in the presence or absence of oxygen, and obligate anaerobes, bacteria that can only grow in the absence of oxygen. Classifying bacteria is important because it helps us understand their characteristics, behavior, and relationship to other organisms. Bacteria can be classified based on several different factors, nutritional requirements, and genetics also. Nutritional-based classification. Bacteria can also be classified based on their nutritional requirements, which include phototrophic, using light for energy, chemotrophic, using chemicals for energy, and heterotrophic, using organic matter for energy. Phototrophic bacteria are able to produce their own energy through photosynthesis and are typically found in environments with high levels of light, such as shallow water or on the surface of plants. Chemotrophic bacteria use chemicals for energy and are found in environments with limited light, such as deep water or underground. Heterotrophic bacteria use organic matter for energy and are found in a wide range of environments, including soil and water. Genetic-based classification. Bacteria can also be classified based on their genetic characteristics, such as the presence or absence of certain genes, their DNA sequence, or the type of metabolic processes they can perform. This type of classification helps us understand the evolution and relationships between different bacteria and their role in the environment. Bacteria can have both useful and harmful effects on our health and the environment. Understanding the benefits and risks of bacteria is important for promoting public health and maintaining a healthy environment. Useful effects of bacteria. Food production. Bacteria play a key role in food production, including the production of yogurt, cheese, and pickles. They can ferment sugars and other organic compounds to produce lactic acid, which lowers the pH of food and helps preserve it. Bacteria are also used in the production of fermented drinks, such as beer and wine, where they break down sugars to produce alcohol. Soil fertility. Bacteria are important for soil fertility, as they break down organic matter and release nutrients that plants can absorb. This helps improve soil structure and promotes plant growth. Bioremediation. Bacteria can be used to clean up environmental pollutants, such as oil spills and toxic chemicals. Some bacteria are able to break down these pollutants, reducing their harmful effects on the environment. Antibiotic production. Bacteria are used to produce many important antibiotics, such as penicillin, that are used to treat bacterial infections. These antibiotics are crucial for maintaining public health and preventing the spread of infectious diseases. Biotechnology. Bacteria are also used in biotechnology, where they are engineered to produce important proteins, such as insulin, and to clean up pollutants in industrial processes. Harmful effects of bacteria. Diseases. Some bacteria are pathogens, meaning that they can cause diseases in other organisms. These diseases can range from mild infections, such as strep throat, to more severe infections, such as tuberculosis. Bacterial diseases can be spread through contaminated food, water, or close contact with an infected person. Food contamination. Bacteria can contaminate food and water, leading to outbreaks of foodborne illness and waterborne diseases. Contamination can also occur in medical settings, leading to the spread of infections from one patient to another. Antibiotic resistance. The widespread use of antibiotics has led to the development of antibiotic-resistant bacteria, which are a major threat to public health. These bacteria can cause infections that are difficult to treat, which can lead to serious health problems and even death. Environmental pollution. Bacteria can contribute to environmental pollution by breaking down organic matter and producing harmful byproducts, such as methane and other greenhouse gases. They can also contribute to eutrophication, which is the excessive growth of algae in bodies of water due to high levels of nutrients. Thank you for watching this video and we hope you learned something new today.